Today we are going to start with the second step of the protein synthesis called translation. As we have seen until now the transcription, which is the copying of DNA into messenger RNA inside the nucleus. Today we will see the translation, which is the change of messenger RNA into amino acid sequence, and this happens inside the cytoplasm. So today we will study the second step of protein synthesis called translation. فإذا we are going to translate. Okay, we are going to translate the messenger RNA sequence into what? Into an amino acid sequence. And as you all know from before, that any change in one of the amino acid can cause the whole protein to be inactive or to become non-functional. Okay, yalla. Who wants to start reading? Who wants to start reading?
تبلش أنا؟
تفضل يا إيمان.
Okay. من الcytoplasm.
Okay, up cytoplasm we have the nucleus.
Okay, in nucleus, then a transcription happen.
Yes.
So single-stranded messenger RNA.
While DNA is double-stranded, فإذا نحن we copy only one strand.
Yes.
Okay. Now single-stranded messenger RNA.
Translation.
By translation.
We have an amino acid chain.
We have an amino acid chain. Now, Ya Iman, we will translate the language of AUCG in messenger RNA into what? Into the amino acid sequence. Now we know we have 20 amino acids, but yes, we have only 4 bases AUCG in messenger RNA. The question now is how this, okay, AUCG, okay, are going to be transformed out, translated into an amino acid sequence. That's what we will see. Yalla, yes. Let's continue. Pose the problem. What is the pose the problem? As you can see, it is a question.
Yes, what are the tools that should be available in order for translation to occur?
Yes, what are the tools? What do I need in order to translate the amino acid, the messenger RNA into an amino acid sequence? Yes, can you continue?
Okay, tools of translation. Document yes. A shows the first tool. It's the ribosome.
The first tool is the ribosome. So it is a small yes. structure found in the cytoplasm, will be needed to convert messenger RNA into amino acid. Yes, continue.
It consists of 2 subunits yes. and 2 sites, chairs inside the large subunit.
فإذا it is 2 parts, a large part, large subunit, small part, small subunit. Okay.
Document B.
Okay, now we go down into the second tool. Document B, the second tool will be what?
Shows the second tool is the translator or transfer RNA.
فإذا now يا إيمان you tell me this is the translator. يعني this is the yes. one who is going to translate from messenger RNA into amino acid. فإذا this one is called what? tRNA. فإذا now نحن yes. we have 2 types of RNA. We have the M messenger RNA and T translator RNA or transfer RNA. Okay.
This translator RNA can read and understand the messenger RNA nitrogenous bases language, ACU.
Okay.
And it translates it into the amino acid language in order to form the protein.
فإذا okay, because of that
tRNA is considered as an adapter.
تمام. Okay, you know what is an adapter? Let's say you have now a new machine in the house. You want to put it in the electricity, but its plug doesn't work. So you get an adapter. الادابتر تحويله.
Yes.
Okay, تحويله مشان يزبط الفيش. Okay, here, this one, the translator, from one side it binds to the messenger RNA, and from the other side it carries an amino acid, let's say amino acid molecule. طيب هلا we will see how it works. Okay. Now document C, the synthesis in vitro. يعني we are trying to do it, okay, outside the cell. نحن we are trying to do it outside the cell. Okay, we are trying to do it outside the cell to see what we can get. يعني trying to get some clues or some information about this process. طيب. What do we mix, Iman, in the beginning?
Ribosome.
Yes.
A translator RNA and the messenger RNA.
فإذا I need these. The ribosome. The ribosome is the one that builds the amino acid sequence. A tRNA is the translator. And messenger RNA is the message that I will use. Okay, that I will use. When I put them together, what I get? For example, you make, ya Iman, an amino acid, a messenger RNA containing you only. يعني you, only you.
Yes.
Okay, look down in the figure below it. Look down in the figure below it.
Yeah.
When you put this messenger RNA containing all you, they call it poly-U. Poly يعني many. With the ribosome and tRNA, what amino acid you get?
PHE.
Phenylalanine, PHE. This means that the U's are translated into PHE.
Yes.
طب what happens when you put A together?
LYS.
طيب C.
PRO.
You see? فإذا now we start learning, okay, that these nucleotides can give these amino acids. Okay? طيب now, let's go back to the table up and please write with me. Okay, what will be the indication here? The indication will be that, okay, mRNA codes for the amino acids. mRNA codes for the amino acids. لأنه when you change the messenger RNA, you get different amino acid sequence. So the sequence now depends on what is written in the messenger RNA. What is written in the messenger RNA? طب, what will be the conclusion here? Conclusion, it will be that protein synthesis depends on the mRNA sequence. So, what is the sequence of the messenger RNA? This will produce for me a specific amino acid sequence or protein. Okay? So, this is very important. This is very important. طيب, now, how am I going to convert 4 letters on messenger RNA, A, U, C, G, into 20 amino acids? You remember when I told you the story about the copying? The story about the copying of or making cars? We said we make a copy and it will be written in English. But the
Yes.
workers do not understand English. We get a translator for them.
Yeah.
Okay, here the same. Here, every 3 letters on messenger RNA are used as one word to give one amino acid. I will repeat. Every 3 letters on messenger RNA will be used as one word translated into one amino acid. Okay? So now I will ask all of you this question. If I have a messenger RNA made of 21 nucleotides, how many amino acids I expect to get? Yalla, everyone, try to answer. Okay? 21 21 nucleotides on messenger RNA. Damn, every 3 nucleotides will be read as one amino acid. Okay, every 3 nucleotides will be used or will be translated into one amino acid. So, yes, Yadam, exactly. فإذا every 3 amino acid, every 3 nucleotides will be translated into one amino acid. Is it clear? Is it clear? So, 30 nucleotides, I get 10 amino acids, and so on. So that's why the scientists make a table. Look at this table. It is very important and we are going to use it a lot. Akid, we will not memorize it, but we have to learn how to use it. Okay. فإذا since we said every 3 nucleotides will be used as one amino acid, so let's see what are these 3 letters as nucleotides. We look first on the left side at the first letter. On the left side at the first letter. It could be U, C, A, G. The second letter will be up, also U, C, A, G. The third letter on the left, كمان U, C, A, G. طيب, let me ask you and let's see if you learn how to read it. If the 3 nucleotides together are G, U, A, what amino acid I get? Yalla, G, U, A, what amino acid I get? Try to look in the table and tell me G, U, A, what will give me? G, U, A, what will give me? Dam said Val or Valine. Is it correct? Others? طيب خالد أحمد Khadija, Hadi, Yusuf. Do you agree? Do you agree, others? Khalid, Ahmed. What if it is CGA? Yalla. CGA. CGA. برافو يا آية، برافو يا دام. أرجينين. اوكي طيب AUG AUG met. برافو يا يوسف، برافو يا آية، دام، Iman, very good. Type. Now, let's learn this once and for all. As they say, every amino acid sequence should start with met. Every amino acid sequence, I will write it down for you. Every amino acid sequence should start with met. Like every letter you started at the top, for example, with a title. Okay, every letter you started with a title. So every amino acid sequence should start with met. This means every M RNA sequence should start with AUG. Okay? Every amino acid sequence should start with AUG. Okay? Very good. Type. Now, I want all of you to look at UAA, UGA, UAG. I will repeat them. UAA, UAG, UGA. These are called what? Stop. Are called stop. What does it mean by stop? يعني the message is finished. Those, they do not give any amino acid. Why? لأنه those are the sign that the message is finished. Okay? The message is finished. طيب, is it clear now, until now, for everyone? Now, I know what is the sequence of messenger RNA is going to be translated to. Every 3 nucleotides will be one word. I will name what they call this word. Third, I have always met in the beginning. It's called the start. And I have at the end one of the three, UAA or UAG or UGA. This is called the stop. Clear, guys, everyone? Please, the others, I want to hear from you. Is it clear? Good يا آية. Others يلا. هادي. طيب يوسف خديجة وأحمد وخالد. يوسف أحمد خالد خديجة. You did not say anything. Where is Khalid? Khalid, you agree with us? Okay, good. Good. طيب, now let us see a video about the process and then we continue reading about it. Okay, let's see a video about the process, then we continue reading about it. طيب. The instructions to make proteins are contained in our DNA like a gene. Now we're starting from the transcription, from the beginning. Okay, now I will make a messenger RNA molecule using which enzyme? RNA polymerase. Okay, I want to make a protein, a sequence of amino acids. Let's see now the transcription. During the transcription, I am building messenger RNA from the transcribed strand only. And now I will do translation after this. Hala, we will see translation, how it happens. First, this is the DNA. Okay, now this is the RNA polymerase, as you all know it. So there is a beginning site here, so it will start from it. And it will use the free nucleotides here to build a new messenger RNA. It opens the DNA and starts copying. But it will use A, C, G, and U. There is no T. Yes, the U is opposite to A. Okay, now this is the messenger RNA. Now the red line is the messenger RNA, and this process happens inside the nucleus. Tamam. Now this messenger RNA will leave the nucleus into the cytoplasm now to be translated with the ribosomes. These are the nucleotides on the messenger RNA. You see, this is the word I am telling you about. Every 3 letters are considered as one word. Now you tell me, the scientists call this word as what? They call it what? They call it codon. They call it codon. Okay? فإذا every 3 letters on messenger RNA are called codon. طيب. Now, this is the table. This is the table. As you can see, the met is called the start. AUG met the start always. And UAA, UGA, UAG is called the stop. Stop, stop, stop. Okay? Stop. Type. Now, okay, the start always AUG and the stop is always UGA, UAG, UAA. طيب. Now, wait. The orange part is the small part of the ribosome. لأنه نحن we said the ribosome is made of two parts. A small part, subunit, and a large subunit. فإذا first, the small subunit binds to the messenger RNA. Now, this is the transfer. The blue is the transfer RNA carrying on its top what? An amino acid. Carrying an amino acid. This is the transfer RNA. This is the anti-codon, the one that is going to bind the codon. AUG on messenger RNA, its opposite is what? UAC. فإذا the transfer RNA carrying UAC will always carry on its top what? The met or the start amino acid. Okay. This is now the large subunit of the messenger RNA of the ribosome. It binds here. Large subunit. Okay. Now we want to take the next codon, the next three letters. Depending on what are the nucleotides of the codon, a transfer RNA binds. Anticodon, okay, it binds the second one. فإذا now I have two transfer RNA, each one binding to a codon, and each one is carrying a specific amino acid. What happens next? The first amino acid binds to the second, and the empty tRNA will leave. A third one now comes. Okay. Now the stop codon, there is no amino acid here. Now the polypeptide chain is finished. The amino acid sequence is finished. Now we stop doing everything and the messenger RNA is broken down or is dissolved. So this is the protein or the sequence of amino acids that I need at the end. Okay? Is this clear, guys? This is the link now. Type. Now, everybody understand the process, how it happens? This is a very important process. Okay, what about the others? Hadi Yusuf Dam, Ahmed, Khadija, what about you? You get it? So you can watch the link again. I put the video, I put the link for you. طيب. فإذا since now, let's read it. Now we have seen it as a video. Now let us read it. يلا يا دام, can you read it please?
Yes, Ustad.
Please, thank you. Okay, now mechanism of the translation. طيب, stage 1 يا دام. It is called what?
استسمعنا.
لا, did you say anything before? I didn't hear a word.
لا بس لأنه صار عندي poor connection.
Oh, no problem. Okay, let's start لكن.
Yeah. The first tRNA.
Okay.
صحن.
The first,
Bind
yes, now we are
always at
starting, yeah, Dan, sorry, with the stage 1 initiation of the start, يعني the beginning of the translation process.
Yes.
Okay, the first tRNA
Bind always at the initiation.
Initiation.
Codon which is AUG. It codes from methionine amino acid. After that, the ribosome subunits assemble to handle both messenger RNA and tRNA.
فإذا always,
We
yeah,
noted
Dan,
that.
sorry, always you start with the AUG on
Yes.
messenger RNA, and the tRNA that binds to it always carry the the methionine.
Yes.
Okay,
Yes.
be noted.
We noted that messenger RNA should be produced from the template transcript non-coding DNA strand and not from the coding non-template non-transcript strand.
Transcribed strand. Okay,
For
this
this
reason,
if the DNA coding strand was given in the exam, then you have to transform it into its complementary template strand before transforming it into its complementary RNA strand.
type from this box, what we study the first sentence, يعني the first orange box, the 2 lines at the beginning, okay? For everyone. From this box, the initiation, we study from the word, the first tRNA, and we end with tRNA. Okay? First 2 lines.
Yes.
طيب. يلا يا دام. The next one, elongation.
Yes. Stage 2 elongation.
Okay.
Ribosome
Translocates
itself forward by one codon,
which is a triplet or 3 mRNA
translocates
base.
itself to. Yes.
Okay.
In this case,
Yeah.
tRNA at T site will be released out from the black, and the tRNA at A site will move to the P.
To the P site, the 2 amino acids will be attached by peptide bond, and the process will be repeated continuously until step 3. As an elongation, now you are making the sequence of the amino acids by binding different tRNA. Okay, to know what kind of amino acid will be coded by each codon, you have to use the table page 80 or 98. It is not for study, it will be given in the exam. For this box, what do we study? We study the part in yellow. Okay, the part in yellow. And the last step, which is termination, at the end, ribosome will reach a stop codon, UAA, UGA, UAG. No tRNA will bind to any of these codon, so the ribosome will be released, the new peptide is produced. Okay, the first amino acid, methionine, will be cut away. It is just used to start translation. فإذن here we finish with the termination stage. For this one, we study the whole box. Okay, it's a small one. The whole box. فإذن translation is 3 stages. Initiation, beginning, elongation, you make the long chain, and termination when you stop releasing the amino acid sequence. Clear? Clear? No problem, Yadam, no problem. It happens with all of us. Okay. فإذن others, the process, is it clear? فإذن now we finish the protein synthesis. We started with transcription, then we move on to translation. By this, I have a sequence of amino acids that form different proteins. As you have seen, the sequence of amino acids depend on the messenger RNA. The messenger RNA depends on the DNA. So this is how the protein sequence, the amino acid sequence, depend on DNA. Okay, this is how the relation between them. Okay, the relation between them. Okay, any question? Okay, let's start with this application. Whatever we finish, okay, we start with this. What is it is left? You continue it at home and we correct it next time. Yalla, who wants to read? يلا يا خالد تفضل.
Research for the role of ribosomes in translation and in vitro synthesis of proteins is done in 2 media.
Okay.
Both media contain
Mitochondrial enzymes,
tRNA, mRNA of
uracil UUUU, and molecules of radioactive amino acid phenylalanine.
One of the 2 media contains ribosomes while the other does not. The intensity of radioactivity is measured in both media. The results are shown in the following table.
Okay. فإذن يا Khalid, we're doing an experiment. Okay, and we're trying to see the presence of ribosome. فإذا we make two media, one with ribosome, one without. So what is the variable factor يا خالد? Variable يعني what is the change between the two experiments?
radioactivity.
لا يعني أنت what you put here and you didn't put here. Ribosome. فإذا it will be now the presence of ribosomes. The presence of ribosomes. What is the posed problem? You want to know what?
Role of ribosomes.
What is the role of ribosomes? I know questions in translation with a question mark. Okay? Type. Okay, guys, listen. I will stop here. I will leave it for you, so please, you read it. Okay, continue it, and next time we correct it. Okay? طيب يلا. Any question before we leave? يلا. السلام عليكم.